Hi, I'm Rodger Tiedemann. I'm a hematologist currently practicing at Auckland Hospital and running a lab at the University of Auckland. I also run a lab at Princess Margaret Cancer Centre in Toronto, where I previously worked for 13 years. And today I gave a presentation at the American Society of Hematology on the cell of origin of multiple myeloma. So in my presentation, I reviewed evidence showing that IgH translocations in myeloma largely arise within germinal center B cells, and that we know that because a lot of those translocations map to immunoglobulin switch regions, and it's within the germinal center that B cells undergo immunoglobulin switch rearrangement. So it's likely in those cells that those translocations arise. There are a number of other translocations in myeloma that also arise from somatic hypermutation or receptor revision, and those are processes that also occur in germinal center B cells. And then there are other translocations in myeloma that arise as a result of VDJ recombination, and those processes occur in cells prior to entering the germinal center, actually in pro-B cells. And all of the progeny of these cells are likely to carry those translocations. Then I reviewed data looking at single nucleotide variations or point mutations in myeloma, and these break down into two groups, those caused by AID and those caused by non-AID processes. AID is an enzyme that's active within the germinal center during somatic hypermutation and immunoglobulin class rearrangement. And it turns out that most of the AID-driven mutations in myeloma are clonal, whereas most of the non-AID mutations in myeloma are non-clonal and so not present in all myeloma cells. And so putting all of that together, the cell of origin of myeloma is often a germinal center B cell where translocations arise and where AID-driven single nucleotide variations arise. And then the germinal center outputs both plasma cells and memory B cells. And plasma cells are often thought to be the cellular origin of myeloma, but we believe that the B cells that come out of the germinal center also carry all of those clonal mutations that are present in myeloma. And we've gone looking for evidence for myeloma B cells and pre-plasma cells that carry the chromosomal translocations, the copy number variations, and the single nucleotide variations of the myeloma plasma cell clone, and have been able to identify those cells. So we've identified cells that lack CD38 and lack CD138, and that are sometimes CD20 positive, that lack plasma cell transcription factors, but which carry all of the chromosomal copy number changes and translocations present in myeloma plasma cells. And so we've shown that in a number of different types of myeloma, and find that across all types of myeloma we can see these progenitor cells, myeloma progenitor cells, but hyperdiploid myelomas do have a lower frequency of these progenitors than other myeloma subtypes. And when we look at those myeloma progenitor cells, they express less plasma cell transcription factors, less IRF4, less NIC, less XBP1S. They're also less proliferative. They express less of the oncogene that may be driven by an IgH translocation. And they also produce less immunoglobulin, they're less secretory. So we can see those cells by single cell RNA sequencing. We can look within, we've clustered a number of myeloma samples together, the B cells and the plasma cells, and we can see this transition from the B cell compartment to the plasma cell compartment. And within the B cell compartment, we can identify rare myeloma B cells that have the B cell receptor of their clone and that also carry the copy number variations of their clone. So they're really a tumor cell, but with a gene expression program that resembles a B cell. And we also see the same thing in an immature plasma cell compartment. We can look at the differential gene expression of those myeloma B cells and the pre-plasma cells, and we can see that the myeloma B cells express some genes that might be expressed by normal B cells. They also express some genes that might be expressed by myeloma plasma cells, but not all of those genes. So they're sort of an intermediate category. And in particular, they express less XBP1S target genes than the myeloma plasma cells do, so they're under less ER stress. And they express more HLA class 2 genes than do myeloma plasma cells, which typically don't express those genes. If we do gene set enrichment analysis to compare those myeloma progenitor cells with myeloma plasma cells, we see that they're less secretory, they have less protein secretion and less unfolded protein response, and they have more B cell differentiation genes being expressed. And this fits with our earlier publication in Cancer Cell in 2013, showing that the persister cells after proteasome inhibition are these low UPR cells, B cells, and pre-plasma cells that lack plasma cell markers and that have lower amounts of XBP1S and lower amounts of CHOP. They're under less ER stress. And other researchers have found similar types of cells persisting after BCMA targeted therapy and after combination chemotherapies.